Today we're going to talk about a subject that is near and dear to my heart, and that is the existence of cell-directed mutations. This is a subject that is the source of a lot of misunderstanding and misconceptions, both by specialists and non-specialists. And hopefully we'll get and cover all the misunderstandings today and help people understand what's really going on and what it all means. So, first, an introduction. Let's talk about what cell-directed mutations are. So, first of all, a mutation. The word mutation, for those who aren't specialists, a mutation is any change in DNA. So it doesn't matter. A lot of people, when they hear the word mutation, they think about, the derogatory term mutant or something like that. The word mutation does not necessarily indicate that something's bad or wrong. It just means that it's changed. In fact, in information theory, that's the way we use it. We have mutators, which are things that change objects. And so, in the cell, it's similar. A mutation is simply a change in the DNA sequence. It doesn't give any. The word mutation doesn't tell us anything about whether it's good, bad, neutral, anything like that. So that's what a mutation is. But a cell-directed mutation is one where the cell contains information about likely beneficial modifications to its own DNA. In other words, the cell itself is orchestrating mutations for a particular function. Now, biologists are often trained to think that either these don't exist, or if they do exist, they are very rare and very unimportant phenomena. In fact, I've talked to a number of people who are trained cell biologists who are completely unaware of the evidence for this phenomena, and even those who are aware have a bit of a difficulty understanding what it means for them. So hopefully this video will clear up a lot of these misconceptions. But the literature, thankfully, has a lot of clear examples of measurably directed mutational processes. So what I hope you take away from this is, first of all, that there's strong evidence for directed mutations. The second one is that I hope to help you rethink your conception of a directed mutation. Because a lot of people think that when we say directed, that a directed mutation must be always correct, or it must be based on the current selection being applied at the time. Okay. Well, what we'll find is that a mutation can be directed, but also in certain circumstances can also be deleterious for some definitions of those things. And you'll see why as we go along. This isn't just word games. There's actually real reasons for this. But dealing with directed mutations is going to require different modes of thinking. The way that you're trained to think about mutations does not always work when dealing with directed mutations. Okay, and I believe that as we go along, that learning to think about mutations in new ways will open up a wide variety of research projects that were previously just unable to think of just because we're thinking in wrong ways. So I'm going to start off about current widespread beliefs about mutations. Okay, and these are generally misconceptions. Okay. Because I hope to correct these. These are not truths. These are the misunderstandings that I hope to address in this video. So the first misunderstanding is that mutations are a result of the breakdown of cellular processes. So the idea is that when cells get really stressed, they stop being able to copy things correctly, and that's the cause of mutation. That's not true at all. We'll see why this happens, and it actually has nothing to do with cellular breakdown, but is actually a regulated system. The second one is that mutations are haphazard, that they don't follow any pattern that is oriented toward organismal benefit. The third one is that even though mutations may have hotspots and biases, those are largely flukes of biochemistry, and they aren't geared, the hotspots are not geared toward biological function. There are some people, these are usually non-specialists, who say that mutations are always deleterious. And then other people say that that the existence of beneficial mutations prove that evolution is true. And then the last one, a lot of people think that actually classifying as a given mutation as beneficial or deleterious is the best way to classify them. And I would argue that that is not the case. So, how should we understand mutations? So, as I said, a mutation is the change in the DNA sequence and doesn't have anything to do with whether or not the mutation is beneficial or deleterious or anything about the effects of the mutation. Mutations occur in every individual. And my question to you is it possible that mutations are actually a physiological process directed by the cell itself? Okay, so before we go into that, let's look through a list of why mutations are thought to be haphazard. Okay, so the first one is that the first experimental way of introducing mutations was developed in the early 20th century using strong mutagens. And these mutations were not beneficial to the organism. They were just damage. So that's the way that we were able to induce a heritable mutation was by going to some extreme measures which physically damaged an organism and all its offspring. And so it was thought that since this was the only way we knew how to induce mutation, that this was somewhat characteristic of what mutation did. And then some of these mutagens that we found were existed in the cosmos, like X-rays. And so the fact that these mutagens existed in the universe caused a lot of people to think, well, maybe the way that evolution occurred is by these X-rays coming from outer space, impacting somebody's their DNA, making mutation. Sometimes it was good, sometimes it was bad. And then this actually got loose in the popular media as well, and a lot of the superhero comic books are actually based off of this idea. Obviously, nobody thinks that's the way that things happen, but just to kind of give you a history of where some of the more colorful ideas in the culture came from. So the other thing is that DNA is a degenerate code, which means that there are multiple base pair sequences that code for each amino acid. So that means that there isn't a direct way to back template from a protein back to DNA. So kind of some of the early ideas was that, their proteins would you'd have a change in a protein, and when that was good, that would feed back to the DNA. Well, the problem with that is that there's not one specific sequence that the protein will feed back to. And so this became known as the central dogma of biology, that genes make proteins and not the other way around. So then there was the Luria, Delbruck, and Lederberg experiments. And these experiments showed that, at least for fairly large classes of mutations, that even the beneficial mutations actually pre-existed in the population. That is, the mutations occurred before the selection. So, if you had a bacteria and you had a bacteriophage that was going to destroy the bacteria, the mutations that prevented the bacteriophage from performing its task would generally actually occur before the bacteriophage was introduced. And so this indicates that the bacteria, or this indicated to people that the mutation was haphazard, but that the and that's why it occurred before selection and not during selection. So the idea was that if the mutation occurred before selection, then it was just happenstance because the bacteria had no idea what was going to happen in the future. But if the mutation occurred at the time of selection, then that's evidence that the selection itself somehow induced the mutation. So they found that for a large number of mutations, certainly impossible for these experiments to prove it for all of them, but for a large number of them, they found that the mutations happened ahead of time. And so that led a lot of people to think that mutations were haphazard because they were not occurring at the time of selection, but they were occurring beforehand, and then selection was simply filtering. And then another interesting thing is that if you postulate that mutations are haphazard, that allows you to separate teleonomy, which is the purposeful way in which organisms act, from teleology, which is the idea that organisms are imbued with this purpose from an external source. So this was the idea that Ernst Mayr used to separate out the obvious purposes that organisms have in their functioning from any theological purpose. Because prior to that, we don't get a lot of this today, but in earlier generations, many theologians used the idea that organisms had their own individual purposes to tie in to a larger cosmic purpose. And Ernst Mayr coined the term teleonomy to refer to the program that organisms have that generate their own functions and purposes, and that because these were generated by haphazard mutations, then this disconnected the teleonomy of the individual organism from the teleology that theologians were trying to impose on it. The other thing is that most beneficial mutations incur costs elsewhere in the organism. So if the idea is that if it was directed, it should be cost-free. Another reason is that most clinically interesting mutations are deleterious, not beneficial. So the if you're studying mutations, the people who are most likely to be studying them are doctors, and doctors, the ones that they are most interested in, are the ones that are deleterious. And then experimental mutation experiments show that changing arbitrary bases of a protein are generally neutral or deleterious. So if I just happen to just choose a base or an amino acid at random and change the sequence, the outcome is more likely than not to be neutral or deleterious. Neutral theory shows that a plurality of the surviving mutations that occur in nature are neutral. And then this neutrality is used as evidence of haphazardness. Another interesting one is that it takes pretty much zero experiments to confirm or write in the literature that a mutation is haphazard. So if you observe a mutation, you can refer to that as a random or a haphazard mutation without any supporting experimental data whatsoever. Just the fact that it occurred is sufficient to support this in the literature. However, if you want to state the opposite, if you want to say that a mutation came from the cell's own direction, I've looked at several papers that do this, and it generally takes between 5 and 15 separate experiments to establish that a given mutation is cell-directed. So you can see that you've got a bit of an epistemological barrier in one direction, but not the other. Another one comes from information theory, and that is the idea that self-modifying code is bad practice in computer programming, so it must be bad practice in the cell. So in computer programming, you rarely write programs that modify themselves because that can be highly unstable. So it would be unwise for an organism to have a program that modified itself because that would presumably be bad practice as well. And then finally, many biologists specifically use teleological word choices when describing mutations. So, that's even if you don't know the source of a mutation, the word choice is almost always in the random, haphazard, accidental, some sort of teleological word, whether or not that's actually what's happening there. It's kind of like the default word choice is almost always in the haphazard direction. So those are the ones that I've seen people use to argue for the reasons I've seen that cause people to think that mutations are haphazard. If you know of others, post them in the comments. I'd be interested to know of any others. But those, from talking to a lot of people about this, that's generally the list of issues that people have. So one of the big reasons why people don't see directed mutations is because I believe our preconceptions of what they should look like are wrong. So most people think that if a directed mutation exists, it should look like this. It should occur in direct response to a bad situation. So I'm starving, so I'm gonna have a mutation. Okay, it should be highly accurate in a single shot. Okay, and then it should occur directly. So, for example, if there's a new food source, if directed mutations were possible, then I should be able to feel out the new food source using some sort of detector, and then that should tell me exactly what DNA sequence I need, and then that will be inserted and will create the protein I need to eat the food that's available. That's the way that most people think about directed mutation. So now there's an abundance of evidence that we're going to talk about for directed mutation, but you have to be willing to think a little bit differently, and we'll get into that as we go forward. Before we go forward, I want to talk about some facts about mutations that are not actually in dispute. They sound like they're in dispute, but if you actually look at the literature, these are facts that are not in dispute and are kind of at the core of the question. So let's start looking at those. First of all, we're going to talk about whether or not cells can manipulate DNA beneficially, and that is not a disputed position. So let's look at a very simple example. I say simple, the actual process is not simple. It's called VDJ recombination. Okay, and VDJ recombination is the system that produces your antibody genes. Okay, so in this process, your cell will modify its own genome in order to be able to produce antibody genes. Okay, now if it didn't do this modification, your cell would have no antibody producing genes. If it does it wrong, it leads to cancer, and your genome has markers which tell the cell where to cut and paste gene parts in order to make these antibody genes. So those are important. So if you don't have this process, you can't produce antibodies. If you do the process wrong, it leads to cancer, and there are markers in the genome which tell the process how to do it. Okay. So what does this process do? Well, it produces combinatorial effects. See, you need millions, billions, possibly trillions of different antibodies for your body to adequately address the threats to it. But the problem is we don't have billions of genes. Certainly not billions of genes are available just for making antibodies. So the way your cell is organized, what they did is they broke up the antibody into separate parts. Okay, I hope you all remember the story of Eli Whitney and interchangeable parts, where he's able to pick a part from each bucket and put together a new gun just from each part. Okay, so this is like that, except for instead of each bucket having the same part, each bucket has a bucket of parts that have a matching interface. So it's like if I had multiple barrels, multiple triggers, multiple stocks, but all of them were machined so that they would work together. Okay, so your body has 51 V segments, 27 D segments, and 6 J segments. And during B cell development, one part from each box are put together. And so this leads to a huge number of potential genes. And there's also some additional ways that it generates additional diversity in there. But from just a few hundred parts, your body can generate about 1 trillion different genes. Okay, now it doesn't necessarily produce all those, but that's kind of the scale of what it could do. Okay, so how does your body know where these segments are, where to cut them, where to paste them? Well, your DNA contains markers, and they're called RSSs, which stands for recombination signal sequences. It tells the DNA where to cut, and there's a possibility that it may provide metadata about the frequency of usage or some other necessary information about how the different parts get used. But you see below that there's the code for the recombinational signal sequence, and you can see that this is known as repeat DNA. A lot of people use the idea of repetitive DNA being junk. Well, actually, in many cases, the repetitive DNA is actually formatting the code so that proteins know how to act on it. Okay, so this shows you which parts of this are the same, which parts are variable. There's two main proteins that act on this. It's RAG1 and RAG2, and these perform the cut and paste operations. Okay, and so it's important to note that these genes and their corresponding proteins are active in the correct cells. This happens in B cells, doesn't happen in other types of cells. It happens at the right time during development, and it does it in the right way. It recombines according to markers in the cell. Okay, if the system breaks down, it can result in either immune deficiencies or cancers. So if there's a mutation that causes some other part of DNA to look like an RSS sequence, it'll have a recombination that could lead to cancer. Okay, so in short, there should be no theoretical objection to the idea that DNA can manipulate itself beneficially, because it happens every generation. We have DNA that produces these RAG1, RAG2 proteins, and these RAG1, RAG2 proteins modify the DNA at the right time, in the right place, and in the right way to generate your antibody genes. So the idea that DNA can provide the information to modify itself is actually completely not in dispute. This happens in just about every organism. So the second truth is that cells can produce mutations in the correct gene when needed. And that's not in dispute. Now, what's interesting about the previous example with VDJ recombination is that those all happened prior to selection. That is, that those mutations in the DNA, that cut and paste process, was occurring in absence of selection. So it's obvious that the cell was directing the genes. It's obvious that it was directing them in a beneficial way because we need antibodies. But all of that was being generated before we had any antigens to be worried about. So these mutations were happening prior to selection. Here we're going to talk about that cells can produce mutations in the correct gene when needed. And we're going to go back to a system that happens after VDJ recombination, but it's still in the immune system. It's called somatic hypermutation. Okay? And to understand this, So if your body hits an antigen that you don't have an antibody for, and you've never seen it before, your immune system then needs to produce an antibody to fight it off. So in somatic hypermutation, the cells mutate their antibody genes, and only their antibody genes. Okay? Not only do they only mutate their antibody genes, they only mutate the region which attaches to the antigen, not the region that signals the immune system. So to see why this happens, if we had mutations in this, so there's basically two halves to an antibody. One half sticks to the antigen, the other half signals the immune system that, like, hey, there's something wrong here, hey, there's something wrong here. So if there was a mutation in the signaling half, then the antibody wouldn't be helpful because it wouldn't signal that it had caught something. The only place where mutations are beneficial are in the part that attach to the antigen, and that's precisely where the mutations occur. And in fact, there is a piece of non-coding DNA that actually targets this mutational system so that mutations occur in the right place. And this mechanism is even turned on and turned off by the immune system when needed. Okay, so we're generating mutations in the right spot in the genetic code at the right time in response to a need. Okay, so just to kind of give you a feel for this, so your genome's about 3 billion base pairs long. Okay, the antibody gene, modifying an antibody gene, that's probably the smallest possible change that will produce the needed effect without unintended consequences. Okay, it's possible that some other mutation may also fight off the gene, but those are probably either longer or they have more unintended consequences. The antibody gene is pretty direct. The antibody gene is about 2,000 base pairs long. Okay, the part of the gene that's mutated is about 1,000 base pairs long. Okay, and as I mentioned, if the other half of the antibody gene were mutated, it would compromise immune effectiveness, not improve it. Therefore, the mutation system reduces the search space for mutations from 3 billion base pairs to 1,000. Okay, so that's a huge targeting. People have said, well, it's not completely directed because there's still, within that 1,000 base pairs, there's still some randomness to it. I'm like, well, I will agree with you that it is, I forget exactly what the number is, but something like, 0.00001% random. If you will agree with me, that is 99.99999% directed, right? So this reduction in search space is direction, because it's not just, as we've pointed out, it's not just an arbitrary reduction in search space. It's reducing the search space to a biologically meaningful area. So this is function. This is not haphazardness. And what's really funny about this system is that many people use this as evidence for the effectiveness of random mutations. Okay. In fact, this is oftentimes used as a model system of how random or haphazard mutations plus natural selection can lead to improved fitness. So what are the reasons why people point to this as an example of random mutation and natural selection? Well, one of them is that it does not primarily produce beneficial mutations. Now, it produces them a lot more often than if we were searching the whole genome. But it still takes quite a few tries to find a mutation that works, even though we've reduced the search space by a considerable amount, but we still have not reduced it enough so that it gets the mutation right with one try or two tries. It still takes a lot of tries. It just, the number of tries that it takes now is exponentially fewer than what it would have been if it had to search the whole genome. The second thing is that it does not work by physically matching the exterior of the antigen to a protein and then copying the DNA code. That was the original idea of how it was so effective, is that it had this kind of reverse back templating of an antigen, and that turned out to be wrong. And so it was just assumed, well, if that isn't the way it's doing it, then the only other option is that it's random. So because it's not, because it's wrong more often than it's right, even though it's right a whole many orders of magnitude more often than if it was searching the whole genome space, it's considered to be random. Okay. And so it's interesting. There's a website called talk.origins, and they had this article called The Evolution of Improved Fitness that uses this somatic hypermutation mechanism as its model selection. But they fail to tell the reader about the reduction in search space for the mutation. And this is what I usually find, is that the fact that there is negative mutations or non-beneficial mutations that are produced by a system is shown to be evidence that it's a random or haphazard process, but nobody ever talks about the idea that the search space itself is somehow reduced in a biologically meaningful way. It wouldn't matter if the search space was reduced, but the reduction was not correlated with biological function. But as we can see here, the reduction in search space is directly correlated with biological function. Antibodies are precisely the genes that we want to modify, and the part that sticks to the antigen is precisely the part of the gene that we want to modify. So this is a reduction in search space that is directly correlated with the biological function being intended. Okay, so far we've established that modern organisms are fully capable of beneficially manipulating their genome. These manipulations can occur before selection, which is what we had with VDJ recombination, and after selection, which is what we have in somatic hypermutation, or I should say during the selection. These manipulations are mediated both by the structural artifacts of DNA, both repetitive and regulatory elements, and enzymes targeted to them. Okay, so both of these processes have enzymes, which are also coded by DNA, which produce the effect. And these manipulations are turned on and off at the correct times for the organism for them to work. So because of this, we shouldn't have any theoretical problem with proposing directed mutations. Okay, we see them all the time. So if someone says, well, it's not, it just doesn't make sense that DNA would modify itself. No, it makes perfect sense. It happens. It's not even in dispute. The bigger problem is establishing a feedback mechanism to the germline. Okay. However, while this might be a problem in multicellular organisms, establishing a feedback to the germline is not a problem in single-celled organisms. Okay. Now, before we go on to that, I do want to go back over some common objections. So one of them is that, most of these mutations are non-functional. And it's true. But the point is that the ratio has been shifted towards the functional side by many orders of magnitude. That's what makes them directed, is that the ratio of beneficial to deleterious has shifted. And it's shifted significantly. Okay. Another objection is the mutations are still random within this new restricted space. Also true, but irrelevant. If you imagine that there was one single mutation, that we were directed to exactly one mutation, that was the correct mutation. Well, technically, that's still a random choice of one. So the important part is whether or not the mutation space is objectively directed, if there's an objectively detectable shift in mutation frequencies towards functionality. Okay, and then some people say, well, of course there's bias in mutation space. All biochemical operations will have some amount of bias. Again, this is true, but the question being asked is not whether or not there is some bias somewhere. The question is, where is the bias pointing? What is the relationship between mutation and function? Because if the bias is reliably and repeatedly shifted towards function, that's an effect that requires explanation. There's no a priori reason why a biochemical bias should favor rather than disfavor a shift towards function. So, we could have a biochemical bias that shifted it in the favor of non-function. If you say, well, this sequence of base pairs has a tendency to mutate this way, well, there's no a priori reason why that particular style of mutation should be more likely to produce beneficial mutations than non-beneficial mutations. So if you see that you're repeatedly and reliably being set up to shift the mutations to bias them in a positive direction, well, that's an effect that requires explanation. You can't just say, well, all of them are biased. The question is, why is it biased in this particular direction? So then the fourth one, they said, well, your comparison of how functional it is against random chance. But we know that mutations do not follow a uniform random distribution. Therefore, your argument is wrong. So the problem here is that people misunderstand the use of randomness in thinking about mutations. Randomness, the idea is not that we think mutations are random. In fact, the whole point of this is that we think that they're not. The point is that random chance is being used as a measuring stick. We are measuring the bias of mutations compared to no bias. So random mutations, if I think about a uniform random distribution, then I am thinking about what is the a priori relationship between mutations and their functions. Okay, but if there's no a priori reason why mutations should be biased in favor of function. So when you do find them biased in favor of the function, there must be a reason. And so the reason why randomness is a good measuring stick for this is known as the no free lunch theorem. And the no free lunch theorem says that basically the short version is that if there is a search function that is well, let's put it this way: there is no search function that is optimal for all search spaces. So a corollary to that is that if you find a search function that is correlated to a search space, then there must be some reason for it. Okay, and then correspondingly, and kind of another corollary of the no free lunch theorem is that a random search will give you average performance. So that's why it's a good measuring stick, is because we can tell whether or not this search function is performing at an average, above average, or below average rate in comparison to all possible search functions. And so if it is performing better than random, that gives us specific information that this process is biased in a way that requires explanation towards function. So the next item that is not in dispute: directed mutations in single-cell organisms are obviously heritable. So we can argue over the heritability of directed mutations in multicellular organisms, but in single-cell organisms, these mutations are obviously heritable. Okay, and directed mutations in single-celled organisms have been known about for a long time. A classic one is E. coli and beta-glucoside sugars. So normally, E. coli can't process these sugars. It's got genes for this process. They're present, but they're not active. But there's a transposable insertion sequence, a little strip of DNA that can be inserted that activates these genes. Now, E. coli will itself insert the sequence, but only when the following conditions are met. The E. coli is starving, and beta-glucoside sugars are present. The insertion sequence is not inserted in the absence of both of these conditions. Okay, so the cell detects when it needs this particular sequence of DNA, and when it does need it, it provides it. Okay, so single-cell organisms change their genome on purpose. Okay, so historically it was taught that mutation is a breakdown of cellular processes. This is patently untrue. Okay, mutation is an intentional, that is a regulated result of cellular activity. So mutations don't happen when a cell is so distraught that it's unable to replicate correctly. That isn't what happens at all. Instead, when a cell is undergoing stress, see, cells have more than one enzyme to replicate DNA. These are known as DNA polymerases. Okay, the default DNA polymerase is basically an error-free one. It's known as a DNA polymerase 3. However, if the cell gets under stress, it's not that DNA polymerase 3 isn't able to perform its job. It's that the cell actually switches which DNA polymerase it's going to use. It switches from the one that does exact replication to ones that induce mutations. So it's specifically trying to induce mutations at the right time. So it goes from polymerase 3 to polymerase 2, 4, and 5, all of which have different styles of mutations that they tend to induce. Okay, so then what it does is it takes genes which are potentially beneficial, like, so for example, if it's lacking an energy source, it might take its metabolic pathway genes and then amplifies them using these mutating DNA polymerases. So it keeps on copying them over and over and over again, using these mutation-inducing enzymes to try to find one, to try to find a change that will use the new energy source. And then when the stress is taken care of, when it's no longer under stress, it found the right combination, or perhaps maybe some other food gets introduced, the cell then switches back to DNA polymerase 3. So you can see that this is not a process of something accidentally happening in the genome. The cell is actually intentionally switching what it is using for replication. So here there's several teleological markers of this process. First of all, the process gets activated and deactivated when appropriate. Okay. Then there are specific conserved genes that are specifically there for making modifications to DNA. And we know that they're there for this purpose because they get switched on when this is the needed result. Then the likely beneficial targets of this mutational process are amplified using this newly activated mutating DNA. So the places where that probably need the mutation are the ones we're going to copy with these mutation-inducing polymerases. So now, as always, as was the case before, this process will produce mutations that are not necessary. But the process introduces a significant bias towards function. Okay, and then just as a side note, some bacteria even have specific markers for exchanging genes with other bacteria, and these are called uptake signal sequences, which is really cool. So bacteria can actually leave genes around for other genes, which kind of gets into the larger question of whether or not in multicellular organisms there can be a feedback mechanism to the germline, and maybe something similar that bacteria are using to exchange DNA material with each other is being used in multicellular organisms. Okay, now There is some evidence for heritable somatic mutations in multicellular organisms. Okay, I'm not going to harp on this real hard because this isn't very well studied, and where it has been studied, the results are currently very sketchy. And I'm not going to pretend like they aren't. But I will note a few things. First of all, immune systems in both plants and animals show signs of having been expanded by retrotransposition. And this is a set of research that's mostly put on by Blandon and Steele. Started way back with their book, Lamarck's Signature, and they've continued to do work in this area. And so it's possible that, if you have you develop a new antibody, that this can be retrotranscribed. Pieces of it can be retrotranscribed back into RNA that can then be put back into the germline. It's possible. And then retroviruses are known to pick up cellular genetic elements. So if you have a retrovirus, it's possible that they pick up elements from your DNA. They've been known to do that. They're also known to be capable of transmitting these elements back to the germline. So, you have these retroviruses, which are generally viewed to be bad, and, so we have these retroviruses, and they are physically transmitting genes to the germline. Now, most retroviruses are thought of as being bad. However, part of me wonders, when we first learned about bacteria, it was thought that all bacteria were bad and that we needed to get rid of bacteria. And the reason for that is because the bacteria we knew about from clinical data. We knew that the clinical data, we knew about them because something went wrong. So we were able to match this bacteria with this disease. But we now know that somewhere like 99% of all bacteria are great. We want them. They're fantastic. It just so happens that the ones that are medically noticeable are the bad ones. And I kind of wonder if perhaps this similar thing is happening with retroviruses, that the ones that are bad are the ones that are clinically noticeable, but that there are possibly good ones around that are doing this transferring function that we really need, but we just don't know about. So, in summary, the actual facts behind the directed mutation are not in dispute. The cell does contain processes that manipulate its own DNA towards beneficial ends. Cells can produce mutations in the correct gene when needed. These systems are obviously directed because they are heavily biased in the direction of function. This is provably true because damage to the system causes damage to these organisms. And in single-celled organisms, such systems are obviously heritable and thus contribute towards evolution. It has not been proven that directed mutation in multicellular organisms can feed back into evolution, but there's no a priori reason why we should discount it. Evolution is just another cellular process and should be studied as such. Now, there's also sociological reasons that I've found why directed mutations are not more widely recognized. Okay, so part of it is the creation-evolution debate. And so on the side of creation, people who are creationists don't like directed mutations because there's a vested historical interest in the following propositions. One, that mutation is almost always bad, and two, that new function is not possible in organisms. Okay, and then on the other side, arguing against them, evolutionists don't like directed mutations because they have a vested interest in keeping evolution teleological. So anything that gives an idea that evolution has a direction seems to go against their deepest feelings about what evolution should look like. So usually, when there's evidence of a directed mutation, you will find the evolutionists will play up the idea that mutation can cause benefit, but they will ignore and omit the fact that it was directed. Or they'll use dismissive language, such as, of course there are biochemical biases. Okay? On the other hand, creationists will overplay the possible negative side effects of the mutation, or they will use a different word than mutation, like adaptation, to describe what is happening. Therefore, you wind up with neither group really wanting to study directed mutations for their own sake, because it winds up kind of being at odds with some aspect of what they're trying to prove. In addition, there's also some epistemological reasons why directed mutations are not more widely recognized. When people think of random mutation, since the idea can include biochemical bias, as we've mentioned before, there are positive biases and negative biases. So bias in and of itself doesn't prevent the idea of random mutation. There's no particular falsifiable pattern that they can point to in order to test the idea. So if you say, well, this mutation is random, and I say, no, it's not, well, how are we going to decide that, especially for a single mutation? So most people don't have a conception in their head that allows them to say, oh, this mutation isn't actually random. Second of all, when people think of directed mutation, they usually have an overly specific idea of what they're looking for, and therefore it is too easy to falsify. So since people are being too specific when they think about how a mutation would be directed, falsification in that particular way makes people think, falsification of that particular way makes people think that therefore the mutation must be random. So because some particular type of direction has been falsified, the idea is that, well, it just then reverts to being random. Well, that's not the case at all. This method of reasoning will prevent anyone from finding surprising designs in the way that directed mutation operates, because a surprising design will, by definition, have falsified the previous conceptual model of directed mutation. So this is why I like to use just the general concept of reducing the search space in a beneficial direction, because there I'm showing something that is objectively true or false. We can tell biologically whether or not I'm constraining function in a biologically meaningful manner, or constraining mutation in a biologically meaningful manner. And we can tell, whether or not these mutations are constrained, and you can tell if it's biologically meaningful. So we can actually measure it. But on the other hand, I'm not committing to a specific mechanism, because I'm not going to know ahead of looking what the mechanism is going to be. What I can tell is whether or not the mechanism is directed, because if the way that the mutation happens, if the mutations that, so you imagine a bias in a biochemical system. If the bias does not perform better than random, then the bias is just a haphazard biochemical bias. If the bias performs significantly better than random, well, that's an indication that this is actually a cellular function and not just a happenstance bias. So let's go back to our list of why mutation is thought to be haphazard. So we talked about mutations that were introduced experimentally. Well, the reasons for mutation in living cells is different from these external mutagens. And that's the same for cosmic X-rays. It's not that these things don't happen. It's just that when we're talking about the normal cause of mutations in DNA, we're generally talking about cellular processes and not these external X-rays and mutagens. Then we talked about the degenerate code of DNA means there's not a one-to-one back-templating available for converting proteins into DNA. Well, as we've seen, back-templating is not the only or even the best method of mutating DNA. And we talked about how the Luria-Delbruck and Lederberg experiments showed that many mutations arose in the population prior to the stressor instead of in response to it. But as we've seen, mutating ahead of time is beneficial in many circumstances, because if, for example, creating antibodies ahead of time is great for the organism. But we've also seen that some mutations are in response to stressors. So that's an invalid reason. Postulating that mutations are haphazard allows one to separate teleonomy from teleology. Well, that's a philosophical reason. That's not a actual biological reason. Most beneficial mutations incur costs elsewhere in the organism. This is true, but this can be mitigatable depending on, if you think about the mutations in the mutations in the antibodies. Well, having a new antibody probably costs the body something in terms of energy requirements, but it's minimal if it's consistent with the overall design of the body. Then you have most clinically interesting mutations are deleterious. Well, that's a selection bias. This is based on what we look at. This is just like with bacteria, where in the early days it was thought that all bacteria were bad, and then we eventually figured out no, most bacteria is good. We just happen to only see the bad ones because that's what we were looking at clinically. And then you have experimental mutation experiments showing that changing arbitrary bases of a protein are generally neutral or deleterious. But these experiments are not about the mutations which actually occur in nature. So if I do a random selection of mutations across a gene and see which ones are good and bad, that doesn't tell me anything about, if cells actually are directing their mutations, then that distribution is not equal to the distribution that the cells are using. Neutral theory shows that a plurality of surviving mutations that occur in nature are neutral. Well, the observation of present neutrality says nothing about haphazardness. Then we mentioned that it takes 0 experiments to test or confirm in the literature that a mutation is haphazard, but it takes a great deal of them to confirm that they are not. So this is not only selection bias, this is also a distortion because it allows any mutation to be called haphazard, whether it is or not. And then the last one is that code modifying code is bad practice in computer programming, so it must be bad practice in the cell, and that is just improper analogical reasoning. So another source of this discussion, I did a paper for biocomplexity called Evolutionary Teleonomy as a Unifying Principle for the Extended Evolutionary Synthesis. It has some of these ideas, some others, but generally talking about the way that organisms direct their own evolution. So anyway, I've got more to say on this topic, but that's plenty for now. I'll do another slideshow later that kind of tells us how we can start thinking about mutations in a better way when doing biology. So thank you very much. I hope you got a lot out of this.